بسم الله الرحمن الرحيم والصلاة والسلام على رسوله الكريم. معكم أخوكم زيد حوسة. أنا anatomical pathologist from Sydney, Australia. I will be giving you the second lecture in the introduction of medical genetics that will be covering chapter 3, the human genome, gene structure and function. We will set the principal definition of a human genome, including the chromosome, DNA, RNA, and genes. And then we will be covering the principles of how gene expression is regulated, including the transcription, splicing, and chromatin modification. When the genetic information in our genes are expressed, that will result in cellular, morphological, and clinical biochemical traits that we will determine what we call the phenotype of an individual. But we have a dilemma here. We know that the human genome will include about 3 billion letters of digital coding carried on that gene and will determine the phenotype of an individual. However, we discussed in chapter 2 that the current estimate that there are only 20,000 protein coding genes. So how could these 20,000 protein coding genes account for the huge variability of phenotype of an individual? So two main characteristics of gene account for this variability. The first one is that many genes are capable of generating multiple different products, not only one. So these 2,000 human genes, they don't produce only 2,000 proteins, but rather they encode hundreds and thousands of different proteins, what we refer collectively as proteomes. And these proteins, these individual proteins, they do not function by themselves. They form within themselves an integrated network of proteins that work in an integrated fashion, and they regulate each other, which we will touch base on in the next slide. An example of this network is in this picture is the genome here that have blue areas, which are the protein coding genes, but also it has some the red areas here, which are the non-coding RNA. These non-coding RNA, that they will be an environment that will integrate into the process of gene networking and will affect the cell function, and eventually will result in variability in the phenotype of the organism. And that will bring us to the central dogma. In human body or human cell, actually, what we consider a eukaryote. So we have a compartmentalization. We have the DNA that we know that it is residing in the nucleus, and the protein synthesis happens in the cytoplasm. So you need that molecule that links the information, carries it out from the nucleus outside to the cytoplasm. Well, and in the human body we call this an RNA or ribonucleic acid. But in other organisms, just like bacteria or E. coli, you don't have that compartmentalization, so the DNA is not enclosed within the nucleus. So the DNA and RNA molecules are very similar. The subtle differences between them that is the DNA is a double-stranded molecule, but the RNA is a single-stranded molecule. The other differences are that the RNA has a ribose sugar component attached to its base in comparison to the DNA, which has a deoxyribose. And the third thing, the third difference between them is the uracil replaces the thiamine as one of the pyrimidine bases of the RNA. So simply, the process is there is a genetic code on the DNA, and that will need a transcription through what we call a messenger mRNA. So the DNA will direct the synthesis and the sequence of the RNA, and then the RNA will be translated into two proteins. So polypeptides and proteins, and these proteins will be involved in the synthesis and metabolism of more DNA and RNA within the cell. So again, there is a genetic code in the DNA, and this genetic code has to be transported to the cytoplasm to something called ribosomes, where the protein synthesis happens. But how this information is carried, it's carried by what we call a messenger mRNA, which is transported from the nucleus to the cytoplasm, where the RNA sequence is decoded, translated, and determine the sequence of amino acid. The other types of RNA that you need to know that the ribosomes themselves, that they are comprised of proteins which called rRNA, ribosomal RNA, which are structural components of the ribosome that which responsible for protein synthesis eventually. And the last type of RNA that you need to know is what we call a transfer RNA. This provides the molecular link between the code contained in the base sequence of each mRNA and the final protein synthesis or the amino acid sequence that will be produced. Okay, هلا ركزوا معي شوي. هلا we will talk about the gene itself. What constitutes a gene? So these are what are the regions in the gene. So we will start with something called exon. شايفين the purple areas here, these are the exons, which are the segment of the gene that ultimately will determine the amino acids. So these all purple segments will be translated into the proteins later on. Okay, so these areas are separated by areas that we call intron. So these are non-coding regions. These are initially transcribed into RNA in the nucleus, but are not present in the messenger mRNA later on. So they will be what we call spliced out. راح يتقطعوا وراح يطلعوا لبرا. And then in each gene we have what we call a 5-end untranslated region and a 3-end untranslated region that are transcribed but untranslated. So also راح يتقطعوا راح يطلعوا لبرا. Alright, so the primary RNA transcription goes usually from the 5-end to the direction of the 3-end. So we have in the 5-end what we call an upstream segment, which is the genomic DNA that precedes the transcription start site in the 5-end direction, which that includes what we call a promoter region that includes sequence responsible for the proper initiation of transcription. Okay, and then going to the other end, what we call it a downstream sequence. The DNA, this is located towards the 3-end direction, and this is beyond, past the end of the gene. And within, we have like multiple what we call a regulatory elements that are located in either 5 or 3 ends. Which we divide into enhancers, insulators, and locus control. Okay, you don't need to know exact details on this. The last thing I wanted to mention in this, what we call a polyadenylation signal, which is a poly A tail. This is something located here in the 3 untranslated lesion and contains a signal for the addition of a sequence of adenosine residues to end the mature mRNA. Okay, so we have an upstream at the 5-end, downstream at the 3-end. We have a promoter at the 5-end. We have a poly A tail at the 3-end. So it's worth noting that the genes are located throughout the genome, but they tend to cluster. They like to live in families. They like to cluster in certain areas within the chromosome and tend to be sparse in other areas within the chromosome. And many of these genes belong to what we call gene families, which share closely related DNA sequences and encode polypeptides, which are closely related. And on chromosome 11, the two important or two famous examples are the globin, beta-globin gene family and the olfactory receptor gene family. And simply these the first gene is responsible for the proteins that will produce the hemoglobin, and the OR genes are basically responsible for our acute sense of smell that recognize smells and chemicals. And for completion, I have to tell you about something called pseudogenes, which are DNA sequences that are closely resemble known genes, but they are non-functional, and they are generally two types. Either they are non-processed pseudogenes representing dead genes that they were once functional, but now they are not functioning anymore or inactivated, and something what we call processed pseudogenes. And these pseudogenes have been formed not by mutation, but something called retrotransposition and reverse transcription. So it's a generation of a DNA copy from the messenger mRNA that will be finally integrated in a DNA. And we will discuss that in later chapters, inshallah. So, as we have emphasized earlier, that the non-coding RNA genes are important as a regulator of the functions of the cells, I think I have to mention the other types of RNA I haven't mentioned earlier. So we've mentioned earlier the transfer RNA and ribosomal RNA, which plays an important role in the translation in the ribosome. But we have other things called like small nuclear RNA, snRNA, involved in the modification of these ribosomal RNAs. And other long non-coding RNAs family that plays a role in gene regulation, silencing, and human disease. And the last type of RNA is what we call a microRNA, which, very small but very powerful. So they suppress the translation of target genes by binding to their mRNA and regulating the protein production of the target transcription. So they more or less control the activity of the protein coding in the genome. And just to address the importance of the non-coding RNA, that we are aware now of some diseases that are related to the non-coding RNA. I don't think you need to know everything on heart, but just there are some examples, just like progressive hearing loss, for example. It is when the presence of a pathological variant of microRNA. The Prader-Willi syndrome is the deletion of cluster of the snRNA on chromosome 15, for example. And the HELP syndrome, for example, the pregnancy-associated disease called HELP, like the hemolysis, elevated liver enzymes, and low platelets, is a result from an abnormal expression of a long non-coding RNA on chromosome 12. So although they are non-functional, directly functional, but they are important, and abnormalities in them will lead to certain types of diseases. And now I will take you to a journey of how the proteins are produced, or polypeptides are produced from the DNA. So we will follow the process of that gene transcription. To start with the transcription, and then RNA processing and splicing, and then the transport, and then translation and protein assembly. Just know that the first two steps, the transcription and the RNA processing, happens in the nucleus, and then everything will jump into the cytoplasm, into the ribosomes. So through the transport, and then the translation and the protein assembly happens there. So we'll start with the transcription. As we know, the DNA is a double-stranded structure. So one strand that goes from the 5' to the 3' and the other one from the 3' to the 5'. And it will end up producing an RNA sequence from the 5' to the 3', which is the direction of production. And as you can see, this RNA it will be synthesized. The synthesized RNA will be corresponding in both polarity and the base sequence to that strand of DNA that goes from the 5' to the 3'. That's why we call this DNA strand on the top is the coding or the sense DNA strand, and the other strand that goes from the 3' to the 5' to the non-coding or the antisense DNA strand. And the transcription starts by a protein called polymerase II, RNA polymerase II, and that initiated at a starting point towards the 5' and one of the most common promoter sequences that we found in this 5' what we call a Tata box, or like we can call it Sunduk City. Another example, what we call like the CCAT box or the CAT box, صندوق البسة. So these are very common promoter sequences that many but not all genes that they do have. And then the transcription continues through the both introns and exons across the gene, and even beyond the position of the chromosome that eventually corresponds to the end of the mature mRNA. And then a cap will be attached to the 3 end, and usually this is the poly A tail. And this poly A tail appears to increase the stability and result of what we call a polyadenylated RNA. The second step that happens also within the nucleus is the RNA processing and splicing. And in this process, the introns, the non-coding parts of the gene, are removed, and the remaining RNA segment joined together to form one strip of mature mRNA. And we know for sure that this RNA splicing is an integral and important step in this process for the cells, because we know that there are some variants within the conserved sequences in the intron-exon boundaries. So any abnormalities that happens throughout this sequence that will result in a shorter than normal mRNA that will affect the eventual function and phenotype of the cells. And we have to know that there are something called alternate splicing that happens. So it's a primary transcript can follow multiple alternative splicing pathways leading to the synthesis of multiple related but different mRNA. Each have subsequently translated and generate different protein products. And we will touch base on the alternate splicing in later chapters. The third step, as we mentioned earlier, it is the transport, so that messenger mRNA will be transported from the nucleus to the cytoplasm. So most of the action happens in the cytoplasm, where the mRNA is translated and into protein by the action of a variety of short RNA, and that happens within the ribosomes. And the action proteins are the completed polypeptides that is assembled. But also we need to remember that there's lots of rRNA, ribosomal RNA, and several dozens of ribosomal proteins are also produced. And the key to translation is a code that relates specific amino acid to a combination of 3 adjacent bases along the mRNA. Each set of 3 bases constitute a codon, and this is specific for a specific amino acid. And if you know that there is only 20 amino acids, and we know that there are 64 possible codons, so that means that, and if the codon is responsible to production of amino acids, we know that one amino acid can be produced by more than one codon. An example here that leucine, for example, and arginine each are specified by 6 codons rather than 1 codon. However, there are some exceptions like methionine and tryptophan. These 2 examples are amino acids that are produced by a unique, each is produced by a unique codon, for example. And also you need to know that there are some codons that are responsible to, so 3 of them are responsible, called stop codon or a nonsense codon, that are designated to terminate the translation through the mRNA. Looking closer to the translation always starts at an initiator codon called an AUG, which is methionine. The methionine is always like the first encoded or amino terminal amino acid of each polypeptide chain. And here we have a clearer idea of what we mentioned previously, that the tRNA, the transfer tRNA, is responsible, or it is the link between the mRNA and the codon on the mRNA and the amino acid that is associated with this codon. And what we have mentioned earlier about there are three stop codons. So whenever the reading frame reaches a point of UGA, UAA, or UAG, so the translation process will stop, so that um, and then the completed polypeptide is released into the ribosome, which available to begin the synthesis of another protein. And you have to know that these produced proteins they are not linear structures, and they are not 2D structures. Many of these proteins undergo extensive post-translational packaging and processing, and they further adapt to their final functional status. An example here is the heme group. It comprises of 4 subunits of proteins, and you have to know that 2 or more polypeptide chains, products of the same gene or either like different genes, might combine to form a single multi-protein complex, which can be further modified in some proteins to addition of a methyl group or a phosphate group or a carbohydrate group, depending on the specific site and the intended function of that protein. And you have to know, within the 3D genome organization, the chromosomes themselves also are complex, are not linear structures. They have like some compartments called some divide them into an A and B type genomic compartments. But what I want to mention here is something called the topologically associated domains, or TADs. And that happens on the molecular level. The genomic compartments are subdivided into cluster of genomic interactions. So the organization of the TADs between cell types and species is consistent, and these TADs are separated from one another by regions called TAD boundaries. One of the models that we see here is the loop extrusion model, which suggests bringing otherwise distal gene ends to each other, and these elements will affect each other. They will be either enhancers or silencers of each other, and this 3D proximity will target the gene to regulate their expression. Another topic that we should mention now, but we will expand upon later, is the concept of epigenetics, which is the study of heritable changes in cellular function or gene expression that can transmitted from cell to cell or generation to generation as a result of chromatin-based molecular signals. These studies focus on reversible changes in the chromatin landscape as determinants of the gene function rather than the changes of the genome sequence itself. Therefore, the key concept is that the epigenetic mechanisms, they do not alter the underlying DNA sequence. The DNA sequence will remain the same code. They will not alter the basic frame of it, but rather there are some other mechanisms that establish this epigenetic status and maintain them, transmit them, and the most important, or like the most famous ones, are either by DNA methylation, the histone modification, or the histone variants. Another topic to mention is the allelic imbalance. So if we have gene that are present in 2 copies, in the ideal situation we have upon expression, it will be expressed, the final outcome will be a product of each of the gene equally. But this doesn't happen in real life, and most of the times we will have some sort of an allelic imbalance, that one gene will be dominating on each other. And sometimes we have what we call a monoallelic expression, where most of the expressed products will have only material from one gene only rather than the other gene. So these allelic imbalances in gene expression is present in the human body. So, for example, unbalanced expression is present in 5 to 20% of autosomal genes, and that's happens at early embryogenesis pathway. And what we mentioned later on as a monoallelic expression, we have several examples in human body, just as the somatic rearrangement we find in immunoglobulin genes or T cell receptor genes. We have the random allelic silencing or activation we found on olfactory receptors in sensory neurons. The genomic imprinting that we found in the parental germline cases, and we have that an example of monoallelic expression is the X chromosome inactivation, that it is an epigenetic silences of allele of one of X chromosome in females, which happens in most X-linked genes in females. So an example of the monoallelic gene expression is the genetic imprinting or parent of origin imprinting. So we know after fertilization, both male and female embryo have one copy of chromosome of the paternal origin, and one of maternal origin. However, during the oogenesis and spermatogenesis, we notice that the imprints are erased by removal of epigenetics marks, and new imprints are established that's carrying only one monoallelic imprint appropriate for the parent of origin. And the last example I will mention here about the monoallelic expression is the X chromosome inactivation. We know that shortly after the conception, the female embryo, both the paternal and maternally inherited X chromosomes will be active. However, within the first weeks of embryogenesis, one of or the other of X chosen to at random base to become further inactive. Through the series of events involving X inactivation center. That X then becomes inactive, and in that cell, and it forms what we call a bar body, which basically a heterochromatic mass present within the nucleus. And the resulting female embryo is thus a clonal mosaic of two epigenetically determined cell types. One expresses allele from the maternal X, and one expresses allele from the paternal X. And that ratio will vary between females. We reached the end of this lecture. I know it's a heavy lecture. Maybe there's lots of new terms that you have been introduced to in this lecture. However, don't panic. Everything that I mentioned here you will encounter and you will see in further subsequent lectures, inshallah. So if you have any questions, please direct your question to this email, and then our WhatsApp group that we will create for this course. All the best, and we'll see you in the next lecture.